Okay. So what I'll do is as we discussed last time, I was somehow unable to take the lecture last time. So I'll just go through all the 4 weeks lectures in short. Like maybe I'll try and complete within an hour. Possibly, I'll go super fast, and then we'll try and do the questions of all the 4 weeks. We'll do as fast as possible, and then we'll discuss whatever if something you all want to discuss. Okay. So in the first week, what Sir majorly talked was about proteins and gel-based proteomics. So in the first week, Sir majorly talked about basics itself, and then a bit of gel-based proteomics, and how exactly we'll prepare samples to do gel-based proteomics. So in terms of gel-based proteomics, there are two major methods, that is two-dimensional gel electrophoresis and differential gel electrophoresis and mass spec. So these are the two major techniques in terms of gel-based proteomics. There are non-gel based proteomics as well, but in this course you are majorly dealing with gel-based one. So in the first week, what Sir discussed was about introduction of proteins in folding happens, how can we purify proteins, and introduction to proteomics. So in general, what is an amino acid? It has an amine group, it has a carboxyl group. Just a minute. And R variable group. So this R is the one which by which different amino acids are present. So the R group is the one which changes here. H here there is CH3, here there is CH3OH, this CH3, CH3OH. So R group is the one which changes, and because of that we have 20 amino acids, and they can be classified in various ways depending upon the need. It can be either polar, non-polar, or it can be acidic, basic, and neutral. The way we want to classify, we can classify in a different ways. And these are a few examples of it: non-polar, one polar ones, and positively charged, negatively charged, and aromatic ones. And to give a brief overview of how amino acids are in general, they are chiral in nature, and they are non-superimposable one, and they can make mirror images of each other. Because only one group is the one which varies, all these groups remain constant in all the amino acids, so we can make mirror images out of it. Only glycine is an example which cannot be made, because in which all these two groups are same. Like R itself is hydrogen, so in terms of glycine we cannot exactly make that. And there are, you might have heard about L amino acid and D amino acid. That is because of its optical properties, whether they are levorotatory or dextrorotatory. That depends upon the polarized light itself. If it is going towards left, then it is levo. If it is going towards right, it is dextro. And that's how they are being labeled, L or D amino acid. And how can we analyze proteins? So proteins which have tryptophan, tyrosine, and phenylalanine in a higher amount, we can detect those proteins at around 280 nanometers approximately. And because of high content of these specific amino acid, we can determine that whether that specific protein is present, whether protein is present in your sample or not. But your protein should have tryptophan, tyrosine, and phenylalanine in high amount, then only that can be done. For example, if there are some proteins which doesn't have these amino acids in high amount, if only one is present, then detecting proteins via this method is not possible. Then we need to go for some other methods. Similarly, you can classify amino acid as essential, non-essential. Essential are the one which we require and our body cannot produce, whereas non-essentials are the one which our body produces itself. And each amino acid have their they depending upon the pH, they behave differently. They can be either positively charged, either negatively charged, or they can be neutral. And the neutral point is the point at which they cannot move. They are neutral. They are neutrally charged, and that is one specific value which is called as isoelectric point. Then how proteins are made, by when two amino acids comes together, then a dipeptide is formed. Similarly, n number of amino acids will come together by forming a bond called as peptide bond, which happens between amino group of one amino acid and carboxyl group of another amino acid. And this peptide bond has a resonance nature, and like they keep on switching like this. And this bond pattern, like either this are at the same side or they can be at the opposite side. So depending upon that, they are either cis form or trans form. I think majorly most of the proteins are of trans form. So they have some different properties. If they are cis, they are bound in a cis pattern or a trans pattern. And then how can we uh, derive the sequence of an amino acid? And basically relation between amino acid sequence and its conformation. If whatever the sequence is, according to the sequence, its secondary and tertiary structure is made. So there was one of the scientists which tried to determine that by using different types of degrading agents, and try to find that that's how because of the sequence, the conformation is different. So that he explained the role of different compounds which have different effects to amino acid or a protein sequence. This urea or guanidinium chloride, what they do, they are effectively disrupt the non-covalent bonds of the proteins. And similarly, so for example, disulfide bond can be broken by using beta mercaptoethanol. So if there is a protein and if it has disulfide bond, that can be broken using beta mercaptoethanol. So that's how they, if we treat certain amino acid or a protein sequence with these kind of chemicals, so these disulfide bonds be broken. You can see clearly those are broken because of the use of beta mercaptoethanol, and this urea will help you to cut covalent bonds. So similarly, he tried to do different, tried to use different concentration and tried to find how exactly a sequence will change, and that's how he tried to prove that depending upon the sequence, the structure of a amino acid depends. The structure of a protein depends upon the sequence, basically. That's what he tried to prove by using certain chemicals, and tried to show that that the sequence is the one which makes the conformation of the protein. Then similarly, what so conformation in the sense secondary, tertiary, and quaternary. So in terms of secondary, there is alpha helix and beta sheets. And so before that, before explaining about the structures, or just give an overview about what protein is, how it came into picture. It came from Greek word proteos. What is it? Is a linear polymer of amino acids itself. General introduction of proteins, and how what all functions proteins do. They have different functions: transport, messenger, structural support, movement. So all there are different examples as well what Sir gave. And then coming to structure, so linear, the amino acid sequence itself is called as the primary structure of protein. Then secondary structure, if it forms like a helical structure, it's called as alpha helix, and if it pleated structure is called beta pleated structures. And how they are exactly formed, that is a bond between the first amino acid and the fourth amino acid. If they link, that's how they are linked to each other, and alpha helical structure is formed. So if you can see, these are the one which are attracted towards each other, and that's the reason this particular amino acid has again come towards this direction, then going towards this direction in a linear manner. So this specific attraction and bonding is makes the structure of a protein into alpha helical form. And there are some specific angles itself, and it can only be stable at those specific angles. And out of this angle, they are not stable. Similarly, beta sheets, they can be parallel or anti-parallel. And this is one of the example of parallel, indicating both amino acid sequence are going in the same direction, and anti-parallel indicating amino acid sequence are going in an opposite direction. And in this process, they form turns and loops as well. So if I give you an example, so these are called as loops. So going in the same direction, and these are called as turns, like going in the opposite direction. So in this process, basically they form turns and loops of making a beta sheet structure, and certain properties of alpha helical and beta sheets. And as I said, there are certain specific angles at which they are stable, that is, which is phi angle and psi angle. So phi angle is between amino group and carboxyl group, whereas the central carbon group and the central carbon as well as the carboxyl group, there whatever the angle is made with the other amino acid, that's called a psi angle. And these are certain psi angles, and these are certain phi angles. So only these are the angles where a protein is stable, and if a protein wants to make beta sheet structure, these are the angles which makes the sheet stable, and these are the angles where alpha helical structure is stable, and whereas this is left-handed alpha helical is stable. Whereas in these angles, none of the protein can make a stable structure, and they are not present in reality. So this was given by I forgot the name of the scientist. It's Raman. I'm not able to recollect the name of this scientist who gave this phi and psi angles. Yeah, yeah, Ramchandran only. Yeah, yeah. Ramchandran's plot. Yes. So he was the one who actually gave this, found those phi and psi angles where a particular structure is stable, and that's how a protein structure is formed. Then, in terms of tertiary, if you go towards much more complex bonding, like in terms of spatially arranging those specific structures, arrangement of subunits and their interaction. So various interaction, van der Waals, hydrophobic interaction, all kinds of interactions comes into picture when we talk about the tertiary structures. And quaternary comes when two separate sequences comes together and forms the whole protein. Then the quaternary structures comes into picture. Then how we can determine the protein? So this is one of the example of detecting using X-ray source. So X-ray crystallography. So basically, these are crystals of a protein, and this is a method which is used to determine the structure of a protein. X-rays are gone through the crystal of a protein. So initially, you need to make a protein crystallized. Then only you can do this, determine the structure of a protein using X-ray crystallography. And then there is a diffraction. Whatever diffraction is happening on the basis of the crystal present as a sample. So the a pattern being made, and that diffracting pattern is being analyzed by using different softwares, and that's how a protein structure is determined. So primary is the sequence of amino acids, secondary is the folding alpha helical or beta sheets only, whereas tertiary comes into picture where overall folding comes into picture, and quaternary is the one interaction among different subunits. So Gesser certain gave certain examples of how mutations can affect the proteins. So if there is one mutation itself, like GAG has been mutated here, and because of which some other amino acid got attached, and because of which a normal red blood cell becomes sickle cell, and that's called a sickle cell anemia. Similarly, in terms of thalassemia, the few amino acids are omitted, and because of that specific sequence, a person might suffer from beta thalassemia. Similarly, about Parkinson and all other diseases are discussed. So all this discussion is on the basis of protein structure or protein folding, which is being affected. That's the reason this kind of diseases can be formed, can be occurred because of wrong protein folding. That's what's discussed in terms of protein folding and misfolding. Mm, Again, the same example of proteins itself. When a protein gets folded, the non-polar sides are the one are outside, and non-polar amino acids are generally facing towards inside. That's what makes them soluble in water. And when urea treatment is given, there is denaturation. The whole structure is being disturbed. And if you remove the urea, it can reform its structure. So there is only one stable conformation, and there are many unstable conformations. As we discussed, there are only phi and psi angles if they are properly there. If all hydrophobic are internal and all hydrophilic are externally exposed. So there are different parameters. If all those parameters comes into picture, then only a one stable conformation of a protein is present. If they are not followed, even if it is forming some haywire structure, that specific structure will not be stable. That's what about protein folding and misfoldings are discussed, and it's the same thing, like represented in a different manner about the stability of the entropy means the unstableness of anything. Then how does exactly the unfolding and folding happens? Because of there are these are the certain proteins which are responsible for a proper folding of a protein, DNA J, DNA K, group E. These are some proteins which plays role in protein folding and misfolding. So this there was an animation video in the lecture itself, like how DNA J first comes into picture, then DNA K comes with the help of ADP, and whatever is the structure of protein that becomes partially folded because after these interactions, and when grow EL systems comes into picture, a protein gets completely misfolded. So these are the few diseases. Then how can we purify once if anything is expressing certain protein and we want to purify out of it? There are various chromatographic techniques. There is gel permeation, there is affinity chromatography, there is column chromatography. There are a number of different chromatographies available by which we can purify a protein: gel filtration, ion exchange, affinity, HPLC, SCX, RP chromatography, reverse phase chromatography, etc. And we did discuss about it how proteins can uh, be purified, and they all follow a bit of different principles, but the final aim is to purify a protein. In terms of gel filtration, so there are certain beads present in the column, and depending, so smaller proteins goes inside the beads, where a larger proteins directly come from outside. The larger will come outside first, and smaller will come later. Similarly, in terms of ion exchange chromatography, depending upon the charge, the beads are either positively charged or either negatively charged, depending upon what kind of protein you want to purify. If they are positively charged, then the negatively charged proteins initially binds to those beads. Then all the positively charged will wash away. Only negatively charged proteins will be present on the bead. Then you can elute them, and you can collect the protein. That's ion exchange. That's how ion exchange works. In terms of affinity, there are specific tags present to your protein, and those specific tags comes and bind to that affinity part of the column. For example, the nickel NTS. So nickel is the um, one which gets attracted towards the his tag. And so if a protein has a his tag, then those will be binded together, whereas all other proteins will be washed away, and then we can elute the proteins by adding imidazole, which actually has more affinity towards nickel as compared to that of his tag, and that's how his tag proteins will come out. That's how affinity chromatography works. And HPLC depends upon the type of column you are using, type of mobile phase you are using, basically the stationary phase and the mobile phase. You can do it, you can try and focus on if you are having polar mobile phase, then polar molecules will come out first. If you are having non-polar mobile phase, then your non-polar one will come out first. So depending upon that, that's how HPLC will work. Then the tech, as we said, the techniques rely on differential solubility, size of proteins, charge on proteins, and affinity for ligands. That's what I just discussed. And also depending upon mobile phases, stationary phases, etc. So matrix, it contains beads, and according to the technique, the beads will change. And a chemical group is generally attached depending upon that your affinity or non-affinity type of chromatography works. So once is gel filtration, it's also called a gel size exclusion. So as I said, the smaller one will go inside, the bigger one will come first, and smaller one will come out later. That's how gel filtration works. And then you can detect it using at 280 nanometer and try and check whether your protein has come out at that specific location, or else you can use certain protein estimation technique like Bradford or Follin-Lowry, and use that specific technique to quantify your protein, whether whatever fractions you have collected, in those fractions your protein is present or not. So that's how your proteins are detected. The initial eluted are higher molecular weight, the later ones are lower molecular weight in terms of size exclusion. In terms of ion exchange, it depends upon positive and negatively charged amino acids. So these are certain beads. This is for negatively charged, this is for positively charged. So this will help in eluting negatively charged proteins. This will help in positively charged proteins. So that's how if the beads are negative, the positive ones bind with the one and negative will wash away and then we can elute the positive ones. In terms of affinity, there is one specific tag attached to it and the ligand or the protein which can attract towards that tag will bind and whereas all other proteins will get washed away. So these are certain examples of that, like what can be the fusion partner for what ligand. That's how this works. And then you can run the SDS page gel and try to find whether your protein is present in the gel or not. And this is how liquid chromatography or HPLC will work. So reverse phase is the one where your mobile phases are of non-polar one, formic acid and acetonitrile. And specific column will be used which will help to elute polar molecules first. Then there is strong cation exchange chromatography, which is also called as SCX. This is similar to that of ion exchange, but even that is used. And a bit of introduction to proteomics. So proteomics is nothing but study everything about the protein itself. It can be either depending upon the abundance, identity, quantity, or if you want to study expression and assay base, depending upon that, your technique will change. So this is the overview of what omics is. Genomics at the DNA level, epigenomics if any environmental factors coming into picture and changing the DNA level itself. At the RNA level, it's transcriptomics. At protein level, it's proteomics. At the metabolite level, it is metabolomics. Then mass spec is used. So this is just the overview of the mass spec. The how central dogma works. And then in terms of protein, they might have go under post-translation modification. Then how can we estimate proteins, as I just said, Bradford or foreign Lowry? So any specific that specific chemical is used, and that specific chemical reacts for in terms of Bradford. Comacid brilliant blue is the one which reacts with the protein nitrogen, and that because of that it gives a certain bluish color, which can be determined at 595 nanometer. Then in terms of SDS page, we can run the gel. So here the separation is on the basis of molecular weight and everything is negatively charged because of the use of SDS. And the higher molecular weight ones move slowly and the lower ones move faster. Then in terms of isoelectric focusing, as we discussed, proteins have at different pH they have different charge. There is one point at which they don't have any charge, and they will stop moving. So that's what the principle IEF uses, isoelectric focusing. So as soon as you add proteins and if they start to move and they will stop at the point at which they come across a neutral pH or a charge at which or at the pH at which they have a neutral charge. Then overview of 2D itself. So these are overviews. So I think in terms of 2D gel electrophoresis, first isolatory focusing is done, and then uh, after that, that strip itself is placed on SDS page and it is run again. So initially that strip is run, and then uh, SDS page gel is run. And if you are using different types of dyes in your sample, then it becomes differential gel electrophoresis. That's what sir gave an overview in terms of in his week one lecture. I think I'm already half an hour up. I need to be much more quicker. I hope this speed is fine enough to just give a overview of the week one lecture. Yes. Yeah. So similarly, we'll go towards week two and then week three and then week four. Then we need to do certain questions as well. I'll try and cover as much as possible. So in terms of week 2, a bit of system biology was discussed. How generally this is a broad overview of it, because system biology is everywhere. Like we cannot exactly. So it basically, so I tried to relate that topic to proteomics. It can be related to genomics, proteomics, metabolomics everywhere, because we are studying the system ultimately, and in that system we are only studying the protein part. So it's proteomics. Then how exactly samples can be prepared, how they can be analyzed, how protein extractions can be done, and how SGS page can be performed, which is one-dimensional electrophoresis. So this is an overview of omics itself. We just discussed how a history about it, how genomics, proteomics came into picture, how central dogma works, and this is the slide which will also tell you about certain techniques which are gel-free based. So we are only discussing the gel-based ones, SGS-based 2D electrophoresis and DIGE, and what kind of stainings can be done in terms of this method. And there are a number of other methods which can be also used to study proteomics or proteomics techniques. Okay, then just a minute. So in proteomics technique, initially you will collect the blood sample, you will try and extract the protein out of it, then you will do different analysis out of it, like the techniques which in terms of gel-based one as the DIGE or differential electrophoresis or 2DE, and then like those are initial methods basically. You can still have some conclusion out of it, but for better conclusion, you need to do certain other assays like tissue biopsies or microchip assays, etc. for much better conclusions. So this in terms of proteomics technique, it just showed 4 different techniques: proteins microarray, label-free detection, or mass spec, and 2 dimensional electrophoresis. These are few techniques by which proteomics analysis can be done. So what is system biology? It is examination of the biological entity itself as a whole integrated system. Everything inside the system, the whole cell, whatever is there inside the cell, everything you want to study. And so basically the whole, it's a higher branch basically. In that specific system biology branch, all different branches comes into picture. So it's rather than study of its individual characteristics, reactions, and uh, components. So if you want to study one specific type of uh, any disease or something, you will try and study everything related to that, right from the gene, right from the protein, the RNA level, and so that specific study is called system biology study, where you are trying to study each and every perspective of that thing. So these are the ingredients of system biology: genome, protein, RNA, signaling, the metabolomics part, networking part, localization part, different organelles which are present inside the cell, the networks, which how the interaction is happening, each and everything of it, of how generally a cell functions. So levels, different systems basically: respiratory system, gastrointestinal system, reproductive system, immune, cardiovascular system. So broadly they can be divided into these systems basically, and inside the system we will obviously include all of these stuffs inside, because every system will have cells inside involved in it, and every cell will have its genetic, its transcriptomic nature, its intercellular signaling nature, how protein is happening, translation processing nature, how it is coming out, and how it is interacting with the other cells, everything. So that's what how complex system biology is. So it's basically holistic rather than reductionist approach, overall approach. And so this is just to give an overview that like whatever we are studying proteomics, it's just a small part of the whole system biology approach. So just to give that introduction, sir involved this specific part in the course. So it can be either model based, data based. We can study in different approaches basically. This is interaction, and this is system information triangle initially. So these are the major ones out of which you. So this is the holistic approach, then reductionist way of studying it, as small as focused you want to study accordingly. So it's the same thing but given in different formats. So it can be either data based or model based. So these are model based, and technology and experimental will give you data. So it can be either data based or model based, and all everything can be interrelated with each other to make to come up with better conclusions regarding system biology. So different approaches according to the triangle, then understanding the whole system. That's how system biology study happens. Mm, So from different approaches like genomics, proteomics, metabolomics, it requires data set from different approaches, and then once it has the whole data set, then it can give certain the whole give a holistic approach about or holistic perspective about that specific whatever the study was involved in. So obviously, by looking at the way it is extremely challenging, and understanding the dynamics of the biological networks requires modeling, stimulation, and understanding of the whole biology, as well as it will require a bit of mathematical and statistical understanding also to in order to compile all of those everything together and come to a good conclusion out of for that specific system. So these are certain references also sir has given in terms of that. Then about sample preparation and application. So how exactly a sample is prepared? For example, in a cell itself, if any of the protein is getting expressed, you have engineered the cell and you want to see if there is a protein being expressed, or in the blood sample, if you take in the blood itself, if a person is suffering from one of the disease, then that those specific proteins will be present in his blood. If he's not suffering from those diseases, then the in the blood itself that specific protein will not be there. So whatever is the sample, so initially those in terms of bacterial sample, you will try and grow it as much as possible so that you can extract the protein in high amount. Whereas in terms of blood, they are already present in high amount, so there is nothing like growing the culture or something like that. So whatever is the blood sample, you will just take it, you will try to extract that. In order to extract that, first will be disrupting the cell lysis, then trying to protect it from proteolysis. So whatever the protease present inside the cell will also be there, and they will try and kill the protein. So you'll add certain protease inhibitors to it in the lysis buffer itself so that all the proteins are protected. Then you will do sample fractionization. So samples can be from anywhere. It can be from tissue, fluid, cell, organism, anything. And from that specific sample, you can study the global proteomic analysis, or the expression way only, or the targeted way, depending upon what approach or what you want to study. Accordingly, that specific way of sample preparation you need to do, depending upon the type of study. So it involves solubilization first, denaturation, reduction, treatment of sample proteins, and then additionally, if you want to improve the quality of protein extraction, then you may lead to loss of selective protein species. So you need to be careful in terms of sample preparation. I think we sir has took sample preparation topic for almost three lectures, I think so, because sample preparation is the one which is the most important part of your whole experiment. The way you will prepare it, only those particular proteins will be detected, and the way you are extracting the protein, then only that specific protein will be detected. There might be certain proteins, if you use certain chemicals for extracting that, then only those specific proteins are visualized, or else they won't be visualized. So your sample preparation technique is the most important one in terms of any kind of omics technique, may it be genomics, may it be proteomics, may it be metabolomics. Your sample preparation tells everything about your whole technique. What you will be seeing basically. So in terms of protein preparation, initially there's solubilization, preventing them from aggregation, as well as so aggregation in the sense from degrading proteolytic enzyme, everything. So protease inhibitors will be added, and denaturing and reducing all proteins and removing nucleic acid contamination if DNA, RNA are removed, and other contaminants as well. And everything is stored at -20 so that it can be stored for longer time. There are certain examples of how good protein sample preparation is and importance of it. So this is the workflow: cell disruption, protection, sample fractionization, protein extraction, contaminant removal, and quantification. So why we need cell lysis? Because cell proteins are inside the cell, then we want to remove out of the cell. So to isolate proteins from intacted cells, then you want to, because of that, you need to lyse the cell first. Then so after that, so how can you lyse the cell? You can use different ways for lysing. You can use osmotic pressure. You can use different detergents for lysing. You can use enzymatic reactions like cellulase lysozyme for lysing the cell, or you can do just freeze thaw. So freeze thawing in the sense by keeping it at -20, then by keeping it at 50 degree or something, or -80 and 50 degree. So giving cold shock basically by freeze thawing. Because of that cold shock, the cell will burst out and it will get disrupted, and that's how cell lysis can be obtained in terms of freeze thaw. And there are some harsh lysis method like sonication, French press, homogenization, or manual grinding itself. So different ways of lysis, and how gentle or vigorous they are, and what kind of samples are uh, used in terms of what specific lysis method should be used for what type of samples. Then, as I said, protease inhibitors are added. These are few examples of protease inhibitors and their specific roles. Then we do fractionization, that is to isolate groups of proteins or fractions from total proteome, so that uh, you can so whatever is the fraction at which your major protein might be present, only that fraction will be analyzed further and not all the fractions. So fractionization methods. So first is centrifugation, or you can use any type of chromatography for purification. So as I said, so sample fractionization. So it involves purification step itself. One of the initial purification step itself is, in other words, you can call it as fractionization. As you will collect different samples, and you whatever is the fraction at which your protein might be present, only you will take that specific fraction ahead for further analysis. Then in the next lecture about how exactly protein extractions can be done. Then different types of components which can be used, like urea, thiourea, or SDS. So everything, their functions and roles were discussed in the lecture. How what it helps in stabilization, unfolding the proteins, etc. You all can go through that. And similarly, basically for different types of components which can be used for different types of protein extraction depending upon the need. Then DTT, etc. It is used for reductant as a reducing agent. Similarly, beta-mercaptan ethanol is also used. Then certain solubilizing agents, so that it gets solubilized and you can run those samples on isolatory focusing or SDS page. So in the sample itself, so reductant is DTT. There are certain carrier ampholettes. There is detergent. There is chaotropic denaturing agents. So these are denaturing agents. So so initially a protein is intact, but they are denatured so that they can be run on the isolatory focusing or SDS page. So you cannot run the intacted protein on the gel. So if you do that, then uh, uh, the protein might not run effectively. That needs to be linearized. So in order to make it linearized, you will be using all of this stuff so that that specific protein is linearized and it is running in the primary structure form of a protein and not in its secondary, tertiary, or quaternary form. Then about what all you need to add into the sample for preparing, depending upon different sample types. Then and so this is in terms of uh, clinical studies, what all factors you need to consider, like pre-analytical factors. Then biology, intrinsic factors like gender, age, extrinsic factors like what diet, modification. So depending upon that, a person's protein level or metabolic level will change. So depending upon all of this stuff, so all this information needs to be collected uh, so that you when your final analysis, when you know that this person had this specific protein in high amount, that's the reason he was suffering from a particular disease, and you try to find a biomarker. But if you don't know its history, what all how he used to be, what all treatment he used to take, and what all food and stuff he used to eat, and what all social history he has, whether he used to smoke or whether he used to drink alcohol. So all everything factors will affect the whole protein level or metabolite level of a person. So all this history collecting or pre-analytical factors needs to be collected so that at the final stage you are coming into a better conclusion. So how you can store the samples, etc. These work, and after that, while purification itself, if a protein is not getting purified from initial fractionization step, you need to concentrate the protein. For concentrating the protein, one of the method is ammonium sulfate precipitation. Then for so basically you need to precipitate the protein and make it much more concentrated. So in order to precipitate, there are different precipitating agents available. One is ammonium sulfate, one is acetone, one is trichloroacetic acid. So there are different. There is a mixture also like TCA and acetone together. Combination is also used. So there are different ways by which you can precipitate the protein and make protein concentrated, because when you are doing the lysis and stuff, it is in a liquid form, right? It is present in the liquid in the lysis buffer itself, and you want to purify it when even after doing initial fractionization or different chromatographic technique, you are collecting the samples in the lysis buffer itself, and you have a samples in liquid form. Then you need to precipitate the protein. For precipitating the protein, there are different methods. Like you can use acetone, trichloroacetic acid, ammonium sulfate, etc. And then your protein is precipitated, and you can just resuspend it in a very smaller amount so that your protein becomes much more concentrated. Then similarly, in that process itself, you need to remove the contaminants as well made by nucleic acid, polysaccharides, lipids, etc. In the purification process, and whatever salts you have used in terms of keeping the protein stable or whatever buffers, then those needs to be removed by using dialysis. Then how nucleic acid contamination can be removed. All those techniques were discussed, how carbohydrate contaminations can be removed. So these are few things you can use in order for removing this. Similarly for lipids, similarly for ionic detergents. So all this can be the interfering factors in your purified protein, and you need to get rid of those components in order to have a purified protein, and then only you can inject that in the for doing proteomic analysis via mass spec, because mass spec is very much sensitive. It can detect even smaller amount of, even like nanomolar amount or femtomolar amount of any specific salt while doing this preparation, or like salt, I'm saying salts are so smaller molecules, even that can be detected by mass spec. So all this needs to be considered very much effectively, and cannot be neglected at all if you want to go towards mass spec analysis later and try to determine your protein. That's how strong a protein um mass spec is. So you all these steps need to be followed much more thoroughly. And one of the trizol method was discussed. So whatever is the bacterial culture that will be grown, then it will be pelleted down, then it will be lysed. Then this particular bacteria is grown. So instead of this, I think I'll show you a video which will explain you in a better way, which was discussed in the, I think, lecture 9 or something. So trizol method was discussed here. What um, so this is also one of the method by which you can do the purification after lysing the so this is in terms of bacterial cells. If it is producing certain protein, if you want to study the bacterial proteomic level, then the cell is grown. Then after cells are grown, the cells are lysed by using one specific buffer, you can use sonication for lysing the cell. After sonication, whatever is the supernatant, you'll centrifuge it, and whatever is the supernatant, that supernatant will be treated with this trizol method, so that you are extract. So in this supernatant, there is everything. There might be certain DNA contamination, there might be certain RNA contamination, there might be polysaccharides present, there might be lipids present. So if you are in that supernatant, if you want to get rid of all of those, then trizol method is one of the method you can use it. So you need to add trizol and chloroform to it. Then once you add that, the upper layer has, and you need to centrifuge it. Then the upper layer has RNA. Then you can take out the RNA out of it. Then add ethanol. Then that will help in precipitating DNA. Then once that is removed, so DNA and RNA is removed by this method, and then acetone is added, which will help you for precipitating the protein. Then when you centrifuge, the protein will be pelleted down, and the remaining acetone can be removed out, and that specific protein can be resuspended into lysis buffer, and that's how you can get the protein out and get rid of DNA and RNA by using trizol method. Then you can estimate the protein using UV absorption at 280 nanometer, or else you can do certain assays like Lowry assay, BSA assay, or Bradford assay. Then once you have the protein, then you can run the gel using electrophoresis. Then who was the one who discovered that? All those were discussed. A general introduction of one-dimensional electrophoresis was given. How SDS page is performed, and so this SDS page depends upon the molecular weight of the protein. So SDS is the one which gives uh, same charge to the protein. Every all proteins becomes negatively charged because of the SDS, and they get linearized as well. All disulfide bonds. So you use DTT or beta mercaptan or ethanol while doing the sample preparation. That will cleave the disulfide bonds and make it linear. And now you can run the SDS page gel. So how exactly SDS is made? So you need polyacrylamide, you need TEMED, you need APS. APS is ammonium persulfate, and units buffers. So while making that, so TIMET is the one which help in initiating the reaction, and APS will do the cross-linking of the polyacrylamide gel for making it solidified. That's how initially a resolving gel is made, and at a lower concentration, stacking gel is made, where the proteins are loaded. And in the stacking gel, initially the gel runs slowly, and then after that, once they come at the same location, in the resolving gel, they are getting resolved. The proteins will get resolved. So as I said, it has acrylamide and bisacrylamide. Acrylamide is the main matrix. Bisacrylamide helps in the cross-linking, and APS is the initiator of polymerization, whereas CIMET gives free radicals so that it promotes polymerization. And SDS is the one which makes negatively charged, and beta-mercaptoethanol will try and help in making disulfide bonds. That's how proteins get run. The higher molecular weight runs slow, lower ones run faster. And you can use certain unknown known proteins as well, or else you can use the ladder itself, so that you can get to know what is the size of the protein, and you should know the size of your protein of interest. Then you can determine whether your protein is present in the sample or not. Then there is something called as blue native page as well, or you can call it as native page, where SDS is not used. So dyes does not denature the protein, but binds to them in the native state itself. And here the purification is in the native state itself. There are other types like multi-protein complexes identification. You can use BN page electrophoresis, where uh, mobility of MPCs, which is multi-protein complexes determined by negatively charged bound comassive dye. So the dye is the one which is negatively charged, and that's what helps in the separation here. This is the summary. So that's what was explained in terms of lecture 2, week 2 of the course. Then I hope you all are getting it. Can we take a break of like 5-10 minutes or so? Yes. Yeah. Mm. Okay, we'll start again. So in week three, uh, Sir majorly discussed about the main techniques, the two-dimensional electrophoresis and Dij in the week four lecture. So how exactly 2DE works? So to do 2DE, this is the workflow. So initially, the first step is obviously the sample preparation, which we did in week two lecture. Then there is isolatory focusing. Then equilibration of the IPG strip after running the isolatory fluorescence so that it can be used for running the SDS page. Then you will do the second dimension, which is SDS page, and then you will visualize the gel. For visualizing the gel, you need to stain the gel first, and once the staining can be of different ways. Then once staining is done, you need to now you can because of staining you can visualize the gel, and then you will see different spots in the gel itself. Then in order to analyze that gel, then you need to do image analysis. Then while doing image analysis itself, then you will overlay control as well as the diseased one or few different samples together, and then you will do while doing image analysis itself, you will um, overlay those images and try to pick certain spots which is overly expressed or underexpressed, whatever. And then those spots picking will be taken, and those specific spot will be cut, digested, and enzymatic digestion of that specific spot is done. Then it is used as a so that is the purified protein itself. Then it majorly has the protein, but along with that protein, there are certain gel components as well. Then you have to do a bit of purification using that, so that all the gel part is removed and only protein is present, and that protein is then used for mass spec analysis. So this is the overview. This is how two-dimensional electrophoresis isolatory focusing is. There is a pH gradient, and it is run as soon as it reaches to the pI, it will stop, and then that is kept vertically like this, and that is run on an SDS page. So just to understand what PI is, for example, alanine, one of the example of glycine here. So it is positively charged at lower pH, and then it becomes neutral at 5.97, and at higher pH, around 9.60, it becomes negatively charged. So this is the so at lower pH, the hydrogen molecules are more in number, so all the hydrogens are present. So because of extra hydrogen here, it becomes positively charged, and there is a point at which H plus and OH minus are equal. Accordingly, this H goes and becomes OH minus, and that's the reason it gets negatively charged. And this is already present, so it becomes neutral because of plus and minus. And then at higher pH, there is higher number of OH minus ions, and there is no H plus greater in the basic pH, and because of that, at higher pH, it becomes negatively charged. This is how the separation happens. At the neutral point, it stops, and it moves continuously until and unless it reaches the point at which it reaches its isoelectric point. So for doing that, initially that specific gel or the isoelectric strip is rehydrated first. Uh, So this is how the rehydration step is. So after the protein extracted, the rehydrated IBG step is overnight for reswelling the tray at room temperature, and that is done either passively or actively by applying voltage or not applying the voltage. And a mineral oil is added to it so that it becomes rehydrated. And Then about IPT strips itself, how general introduction of it, how much is the length, what is the pH ranges available. So depending upon the need, you can use such kind of IPG strips. Depending upon the size of the strip, how much dye need to be used in order to stain the gel. Then this is what the first dimension is, that is isoelectric focusing. Then once that is run, you will be doing the equilibration. Once the run is finished, you will be doing the equilibration with the for initial buffer. Then with the second equilibration with the second buffer. So this first buffer involves reaction one involves DTT where it breaks the disulfide bond, and the reaction two involves iodoacetamide, which will add certain groups to it so that it doesn't rejoin again. Then in terms of second dimension, so in order to explain this, what I was thinking was I'll go through this lecture 9 video. So this is about sample extraction. I'll just stop this. Uh, okay. So this is how the initial protein extraction is. So these are the steps. You need to harvest the cells. You need to sonicate it and extract the proteins. So you need to inoculate the stains like this. So there is a bacteria inside the plate, and from the plate it is taken into the LB medium. Then you will inoculate. You will incubate at 37 degrees for overnight, and then once you have, so that's how the culture will grow. Once the culture is grown, you will take the sample and centrifuge it. Once you centrifuge it, there will be a pellet which is obtained, and that pellet is resuspended in lysis buffer. And once that is resuspended in the lysis buffer, or you need to wash the pellet so that the media is uh, washed away and there is no media present. And in the lysis buffer, you will sonicate the sample. That's how the sonication probe looks like. During sonication, the cell will be lysed like this. Then these are certain sonication parameters which you need to set. There is certain pulse given. So there is on and off system for giving the pulse. 30 seconds on, 10 seconds off, and it is run for some number of cycles, 5 or 10 cycles, and the total time is being given for 10 minutes or 20 minutes, depending upon the need. Then once the cell is lysed, then it is centrifuged. The supernatant will have everything. Then after from that you need to have only protein. Then trizol method is used. You will add the trizol and chloroform as well. Then after incubating and centrifuging, the upper layer has RNA that will be removed. And after that, then you need to add ethanol. Then after you add ethanol, uh, the DNA will be getting precipitated, and this is how the DNA gets precipitated. Then you'll take the upper layer itself, which will have protein. Then then you will add acetone for precipitation of proteins. Then similar kind of pellet you will see for proteins as well. Then you need to dry that specific pellet and resuspend it in a specific buffer, and then can be used for protein quantification. For protein quantification, one of the method is Bradford assay. So that is, I think, common, which you might have done in your undergrad as well. So I'll not go to the Bradford assay. Then I was explaining about, I think, lecture 11 as IEF. Okay. Okay. So this shows the workflow for isoelectric focusing. I'll just go through that. So this is the animation, but I'll go towards the in the lab protocol one. Yeah. So first step is isoelectric focusing, how it is done. So first is You will do the rehydration of IPG strips and the isotopic focusing of the rehydrated strip itself. So this is how the rehydration happens. In the tray itself, we will add the IPG strip and rehydrate it first with the specific rehydration buffer which we just discussed. So this is how the strip looks. Then you will add the mineral oil to it. Then this is called as focusing trick where your samples are actually run. Then you need to use certain paper wicks as well so that once you keep the strip in the focusing tray, it doesn't move here and there and it is stable enough. Then you add certain water initial for soaking to it. Then whatever the excess mineral oil that is removed first, and then again the strip is kept in the focusing tray, and electrodes are these are electrodes how they look, they are attached, and the paper wicks are also kept like this just after the strip, and electrode is kept so that it will run. So the pH gradient is in the strip itself. Then now you will load the sample, and according to the pH gradient it will move. Then you will adjust certain parameters to it, like what is the pH range, what is exact, all those parameters you will give in the PC, like the software, and then it will run accordingly, something like this. So your sample was loaded, then it will run, then your proteins, whichever, wherever the isoelectric point is, it will stop moving it, whereas the proteins which will continue to move, they will try and find their isoelectric point and stop moving there. So that's how isoelectric focusing happens. And then in terms of once the sample is run, you will equilibrate the solutions with DTT, and so this is how the equilibration happens. You will place the strip inside this tray, and you will add the first equilibration buffer which has DTT in it. Then whatever are the proteins that will be, so if you look at this, so the disulfide bonds which are present in the strip, they will be broken. And in the second step, you will add the buffer here. Then again, you will remove the excess initial equilibration buffer from the tissue, and you will keep it back into the second equilibration buffer, which has uh, uh, iodoacetamide, which will add this group to the sulfate group, and it will doesn't allow the reformation of this disulfide bond, and now it has been equilibrated. Now it can be used for SDS page. So that's what two-dimensional first dimension of the 2D works. Where are you? You all got it right. The initial first dimension as well as the protein extraction. Yes. Yeah. So now we'll move ahead for the second dimension. So this is what, in terms of second dimension, how the casting is been done. So the strip, you are soaking the strip in the tank, and so in the SDS gel will be made first. So in terms of this, initially a resolving gel is made. We did discuss about the components of SDS page. Then those accordingly, those components will be added: resolving gel, then the stacking gel. Then this specific strip which has been equilibrated, then that will be added onto the top of the gel, and a ladder will be loaded so that whatever the proteins present here, they will be resolving here, and after loading the ladder, you will add the another buffer above this specific. So this is the strip you will be adding. So you, if you can see, there is some buffer being added on the top. So the buffer will be added on the top so that this doesn't become dried, and uh, so drying should be avoided basically. And in order to avoid that, another buffer is added onto the top, and then it is placed into the tank buffer, and now it will the second dimension will be running on this, uh, where the samples are present in the strip itself, which was run in the first dimension. Then you will place the gel in the buffer tank, and that's how the separation will take place. The higher molecular one will move slower, the lower one will move faster. Then once you have that second dimension one, then you will now stain the samples, stain the gel. For staining, there are different ways of staining: Comasi blue staining, silver staining. There are a number of different stains. Uh, in terms of staining, what do we do is, if I think if we can just go through the video once. Where is that? It's not in this. So this is how the gel is setup is initially made. Uh, so ensure proper packing. Then the gel is loaded, SDSP, the equilibration strip is kept. So this is how the strip is kept. Then again, once the strip is loaded, you will load the ladder. Then again, you will overlay it with agarose again, so that it doesn't become dried. Then you will keep it in the tank and you will run the gel. Then in terms of staining, this is, I want to show you the actual lab one. These are animated videos. Yeah. So this is how the staining is. You need to remove the gel, do initial fixation, then you need to add the stain application, whatever the stain you want to use, then you need to de-stain it as well. So removing the gel and fixation, the fixation is generally with methanol and acetone. Then this is how the gel is removed. So you need to open the glass frame, uh, detach it from the corner first, and then you just need to slide it onto the uh, this is how the gel looks, and then you need to slide it onto the buffer solution first. Then you need to add the stain to it, application of stain to it. Then you need to revolve it for, like, rotate it for a certain time so that every part of the gel is stained. So this is how the staining happens. Once it is stained, then you need to remove that staining solution and add the de-staining solution to it, so that whatever is the other part of the gel, the surrounding part of the gel, is de-stained, and the proteins which are present, only that specific protein is stained, and surrounding is de-stained. And this was just one example how staining is done in terms of Comasi blue, but there are a number of other stains as well, and everyone has their own benefits, you can say, of using certain type of stain. So most commonly used stain is Comasi stain itself, because it is non-hazardous. So you'll keep the gel inside, and this is how initially you need to do the fixation by using methanol and acetic acid. Then you need to stain. In terms of silver staining, initially the Ag is reduced to Ag ions, and that ions are again, like formaldehyde in the alkaline solution is added in order to reduce silver ions to metallic silver, and the reaction is stopped with the help of acetic acid. Then it is visualized. Similarly, there is cyproruby as well, which detects low molecular weight protein. So if you are interested in low molecular one, then you need to use such kind of staining dyes. Everything has a similar process. Like, you need to fix it, then you need to add the dye, then you need to add the de-staining solution, which has methanol and acetic acid, and then you need to visualize. Then visualization also depends upon the type of staining you are using. Depending upon, accordingly, those specific wavelengths will be used for detecting those specific bands in the illuminator. Similarly, there is Procure diamond. Everything is similar. According to the dye, the emission wavelength will change, and accordingly the appearance of it will change, and the sensitivity of each dye is different. So these are the sensitivity of it. So these dyes can detect even 1 nanogram of protein, whereas these detect about 50 nanogram of the protein. So if it is less than that, this might not detect. This is how what was discussed in terms of lecture 2, lecture week 3 lecture. Then uh, image analysis. So now you have visualized the gel, like you have the image of the gel. Now you need to do the image analysis. That image analysis can be done by different softwares. So these are the images: densitometry or typhoon variable mode imager. uh, so sir discussed about the softwares as well. So just to discuss a bit about software, how exactly that was done. Is it in lecture 13? So now you have the image for two-dimensional electrophoresis. I'll just show you how image analysis was done. Okay, so there are different softwares available for two-dimensional electrophoresis. These are different websites you can use for analysis. So basically, you need to select the two images. Then now, if you want, you can crop certain images as well, whichever is required, and accordingly study those specific proteins only, depending upon the need. And then also you can overlay those images like this, and while overlaying, which are the one which are overlaid, you can identify and you can see that if anything is not, some different proteins are expressed in control group as compared to that of disease group. Accordingly you can do, then you can also do statistical analysis, which has higher intensity, which has lower intensity, which has been visualized into the gel. You need to make sure that even while sample loading also, you are loading same amount, because you have quantified the protein, right, using Bradford assay or Fallin-Lavrie assay. So that's the reason quantification is important. So if you are loading a higher amount in one gel and you are loading lesser amount in another gel, then itself the visualization will change. So you need to make sure that you are loading similar amount. Then you can do certain smoothing, like image editing things, so that, and also it will tell you about the area of intensity of it, the area of it, everything about the protein. Then for making the image proper, you can do certain editing also. It tells you about the intensity, area, volume, everything about the each spot, and it gives spot ID as well. And do certain statistical analysis as well, like coefficient variance, central tendency, everything that can be done inside the software. Then about each spot, it will give you all the details: what's the molecular weight, what's the area of it, what is the volume, what is the intensity of the protein compared to the other gel. So this is what software analysis was. Then this is was some different software which was discussed with the sir. Then you can, so you can take certain different samples. You can select the dye which you used. Then once you select the dye, whatever the samples you have selected, initially the visualization is not accurate enough. Then you will, what you can do is you can change the intensity of it, everything, so that accordingly different parameters needs to be adjusted. Then it can be visualized. Then the smoothing, everything, then all those parameters, minimum area, what you want, if you change accordingly, then uh, the image is getting visualized enough. So that's how visualization occurs. Then once you can visualize this, you can zoom any specific location, or else you can zoom locations for all three images together as well in the software, something like this, which will be shown. So at the same location itself, you can zoom into each and every image at once itself, and you can determine which are certain different proteins, or which is getting highly expressed or upregulated or downregulated. Then you can do certain statistical analysis like this. We'll come to know which protein is highly expressed, which is not that highly expressed, and what is the distance between those proteins, whether it was similar or something different, it is right at the same location, or there is some changes in the uh, molecular weight of the protein, etc. All those can be visualized. Then you can do multiple images like this, then you can compile all of those together as well. Then in that you will get an Excel sheet, the molecular weight, intensity, area, everything of it. Then you can compile. So once you did both this analysis, you can compile these two together as well, and can see all those images at once also, and overlay those images and try to find which one is getting overexpressed or uh, upregulated or downregulated. So you can find the uh, maximum intensity in which type of analysis it was formed. Basically, it will take the average of all these three images. So if there are multiple samples, you can do the averaging as well and try to find how much is the area and how much protein was expressed in what kind of samples. So that's what image analysis software helps you with. So we discussed about all of this. Yeah, after that, sir, I think discussed about certain case study examples, how you can do such kind of studies. You can take control sample, the disease sample, do some same kind of analysis, uh, take the sample, do lysis of it. There's certain protocol for extraction of the protein. Uh, like you need to optimize the protocol, so this protocol works much better for protein extraction. Then after protein precipitation, after extracting, they try to find that using one type of precipitation method, this is how the protein looks, and using other type of crude versus depleted. So depending upon the method, the type of so that's what I was saying. Your sample preparation will change everything, the way you want to look at your results, and also the staining which you use, as because every stain has its own sensitivity. So depending on the amount of protein loaded, so every factor will affect the type. So although everything is the same sample, but the way the sample preparation was done, accordingly the type, the way you can visualize your gel image is different. Then there's a protocol by which, like, you need to have multiple samples. You need to label those samples. What is the healthy one, the diseased one, and you can run that. Then you can try to compare those images using image software analysis and try to come to certain conclusion which are upregulated, which are downregulated. So these are healthy control. These are the diseased ones. So these are lesser than the in the healthy one. So they are down regulated, and as compared to the healthy one, these are up regulated. So that's how these studies are done. Then mass spec analysis are done. So that in order to determine whatever protein you are seeing, what is that specific protein, you can analyze that using mass spec. So they found that these are few proteins which were down regulated. These are few proteins which were up regulated. That's how it can be applied. Basically, whatever you are studying, this is the way by which it gets applied. Then one more example of malaria was given, then how exactly malaria grows. Then once you have that, how can you extract the protein out of it? You will run the gel. Once you run the gel, then how specific drugs have been. So this is another approach by which you can do proteomic analysis. So one specific drug has been given, and because of the presence of that drug, how exactly the person's proteome is getting changed. So they found that these were the few which were upregulated, these were the few which were downregulated with this specific drug, and with this specific drug, all these were upregulated. So altered protein expression levels because of the different drug exposure. Some proteins were upregulated and some were downregulated in different types of drugs which were given. So that's another approach of using proteomic study. So sir explained about different, there was one more example of COVID. So three different case studies were discussed. So this is a good gel when everything goes well. When something goes wrong, this is called a streaking of the gel. This is if such kind of gels you see, it's called as trace impurity. It's because of trace impurity. If you see images like this, it is because of old timid. Then when the whole crude sample is loaded, nothing is being lysed or extracted, whatever. Once you are taking direct the sample directly, that's called as crude sample. Uh, so these are few examples of good and bad gels basically, and what might be the probable reason because of that. So only equilibration with DDT will give you such kind of gels. If you equilibrate with DDT and as well as iodoacetamide, then the image will be different. And this is because of over focusing. So yeah. So these were the different perspectives of uh, what was taught in the week 3 lectures. So I tried to cover a bit of lab work or how exactly it is done, which was asked in the last lecture. I was trying to do that. Then dive week. Let's go for week 4 lectures. So it is also similar to that of 2DE, but here we are using 3 different dyes basically. Just a minute. So in terms of week 4, the other gel-based proteomics technique was discussed, which is dyes. Then further identification using mass spec, using Malditov as a detector. So this to overcome intrinsic gel-to-gel variability, dyes is used because it is multiplexing technology because 3 different dyes are used, which is Psi2, Psi3 and Psi5. So each and every dye has, although they are of similar molecular weight, but they have different excitation and emission. So that's the reason they can be visualized differently. If you give certain wavelength, then inside the gel, although these 3 samples are run together, only those will be visualized which was uh, uh, stained by one of the dye Psi2. And then accordingly, if you change the excitation wavelength, only Psi3 will be seen. If you change it to 620, only Psi5 samples will be seen. So multiple samples can be run in one go and analysis can be also be done together, like the control sample, disease sample can be run together and you can visualize them if they are getting overexpressed or downregulated or upregulated. So it depends. There is 2 types of labeling: minimal labeling, saturation labeling. So in this lysine is used, in this cysteine is used. So why lysine and cysteine? Because of that is how general distribution of proteins is in terms of any. This is an average of it. Like most of the protein has leucine for sure, but there are few proteins only which has cysteine and tryptophan, or the number of proteins, number of these amino acid are lesser in the protein as compared to these proteins. Then lysine labeling, where cyanine dyes react primary amino groups of target proteins, which is lysine. And minimal labeling, only 3% of the whole proteome is labeled. This is how the labeling happens, the structural, how exactly the dye looks like, and where it binds to the protein. It binds to the amino group of the protein. It also consists of dimethyl formamide, this side eyes, and this is what the lysis buffer consists of, and it can label around 50 microgram of protein. So there is control group, there is treatment group, all those are pooled together, which is called as internal standard. So this will basically internal standard will help you to reduce gel to gel variation, gel to gel match, and comparison easier happens. So whatever is being observed in this internal standard, only those specific proteins will be considered. If there is something which is observed, which is not being present in the internal standard, those specific proteins or spots are not considered in your analysis. So there's sample 1, there's sample 2. You will mix them together to make the internal standard. So sample 1 you are mixing with Psi3 dye, sample 2 you are mixing with Psi5 dye, and you have mixed these two together, which is internal standard, that is being uh, uh, stained with Psi2 dye. So now these are the three samples. Maybe this sample 1 is disease 1, sample 2 is the control 1. We'll just mixing them. So this internal, this green spot, when you do the visualization of it, the green will be everywhere because it has both of these samples, sample 1 as well as sample 2 together. Whereas when you are visualizing with the sci-fi dye, only those will be seen. With the sci-3 dye, only those will be seen. So this basically helps in much more better analysis than that of two-dimensional electrophoresis. But using these dyes are little expensive, so uh, if there is good funding enough, then people go for dyes. If not, then people go for 2D 2D dimension two-dimensional gel electrophoresis. So then once you have these three sample mixed with the certain dye, you will mix them together. Now this will be loaded into the gel like this. So now isotopic focusing will be done. So it is similar to that of 2DE, but the initial sample preparation itself has certain difference. So that's the reason it's called as differential gel electrophoresis. So once you run that, then you will do the two-dimensional electrophoresis. So this is what the excitation emission wavelength is, and that's how they look: green, orange, and red. And so by using internal standard, it minimizes gel-to-gel variation, it minimizes biological variation. It can be further analyzed using statistical tools because you have internal standard as well. So you can give the image software itself that this is the internal standard, consider only those in your sample 1 and sample 2 visualization. So accordingly it will give you everything, the image analysis what we have just seen. Similar to that, this image analysis will also work. So this is how the visualization will look like. So once you run the gel, if you give certain excitation wavelength according to Psi3, then these proteins will be visualized. So if you see, there is this protein which is only seen in Psi2 but not in Psi3, whereas Psi5, so internal standard is the green one. So these are the proteins which you can see in both of these two together. So this is the protein which is only present in Psi5 but not in Psi3, whereas this is the protein which is only present in Psi3 but not in Psi5. So that's how you can determine from where these spots are coming into picture, whether it is coming from sample 1 or sample 2, because it has both of them together. So the sir just gave overview of like blood samples are collected, then multiple serums are extracted out of it, then uh, the two-dimensional electrophoresis is performed. Each and everything will be control samples will be given with one dye. Test samples will be stained with one dye. Then all of them mixed together will be stained with certain dye, and they are mixed together. And then the gel is run. Then it is observed in the illuminator, giving different excitation wavelength. Then accordingly, different uh, samples where spots can be observed depending upon the type of excitation you are giving. That's what the overview of dyes is. This is how the gel samples look. Then by using dicer software, you can understand which spot is has been overexpressed, which is uh, down regulated, etc. Then sir gave one of the case study of prostate cancer of PCA protein, whether it got upregulated or downregulated in the cancer patient compared to that of healthy sample. After collecting the sample, it was purified using affinity chromatography. then it was run on the gel. So in terms of this specific cancer, there is a score called as Gleason score. So they took that's the intensity of this specific cancer. Okay, so it is nothing related to the technique. So depending upon that, those samples were collected and those samples were divided into two groups. The one which had Gleason score of 5, they were in one group. The one which had Gleason score of 7, they were in one group. Then accordingly, Dij was performed and mass. So that told us that these are few proteins which are upregulated. These, these are few which got downregulated. So these are the same proteins, PEDF and zinc alpha glycoprotein. The PEDF got upregulated in this specific patients, whereas zinc alpha glycoprotein was similar in both the Gleason score patients. Then, as I said, like if you want to validate it in a much more accurate manner, only this doesn't uh, tell you the exact result. You need to do certain other proteomic analysis like immunohistochemistry. So in this, there is a histome means tissue. There is a tissue. In that tissue, there are certain cells expressing certain proteins. You'll add primary antibody to it. The primary will be attached to that specific protein which is expressed onto the tissue. Then you will wash the cell. Then you will add wash the tissue. Then you'll add the secondary antibody specific to primary one. Then that secondary antibody is attached to an enzyme. Then you'll wash it. Then you will add the substrate, and that reacts with the enzyme. And if the intensity is greater, it indicates that your tissue sample had much more those specific proteins. That's the reason your primary antibody attached to that. And depending upon that intensity, people come to a conclusion that that specific protein is expressed in the tissue. Then mass spec analysis, how exactly that helps. So your protein should be uh, much in a purified form itself. Once you have that, you can inject the protein in mass spec. What does mass spec do? It's a technique for production of charged molecular species and separation by magnetic and electric fields based on mass to charge ratio. So as we said, once you collect that specific spot, you do enzymatic digestion of it. So that spot has the whole protein, right? That protein is now digested so that uh, mass spec can analyze it, because that protein will be high molecular weight or low molecular weight, whatever. But it is in kilodaltons, like 40 kilodalton, 50 kilodalton. So you need to digest that specific protein by using enzymatic digestion. So digestion is specific, right? Like trypsin has some way, like it digests at one specific amino acids itself. Similarly, chymotrypsin has its own way of digesting. So only those kind of specific smaller fragments will be obtained. And if you can detect those smaller fragments in mass spec, you can come to by joining those specific fragments, you can come to a conclusion that that specific protein was present in that specific spot, and that will ultimately indicate that that protein was present in your sample. So mass spec will further confirm that whatever protein you are seeing, that is for sure the protein of interest which you are looking for. So in terms of mass spec, you need to do ionization first, then you do separation using as per the mass to charge ratio. Then you can detect that by using different softwares. So once you have the diage, once you know that these are the proteins which are expressed, then you will cut that specific part of the gel and you will do enzymatic digestion. Then that you will use as your sample. So if there is a sample, so analyte is your sample. Then once the laser beam comes in contact with your sample, it will ionize the sample. So that's what it is called as ionization. And this is the matrix. So it's called as matrix assisted laser desorption. So this is the matrix. It is assisted by, so sample is assisted by that, and the laser is being targeted to the matrix. Then that will ionize the sample. Then once the ionized sample will run towards the top time of flight, which is the analyzer for mass spec. Then certain merits and demerits of it. So this is how the mass spec instrument, you can see this is, so you have a source of ion, then you have the sample. Because of the laser, this specific sample will get ionized, and as per the mass to charge ratio, they will get separated. The smaller ones will move faster, the larger ones will move slower, and they will be then be detected by the detector. So there is some specific formula by which you can determine what is the time of flight. So it resolves on the basis of mass to charge ratio and measures the mass to charge based on time and taken by ants to fly in the analyzer and then strike the detector. So it depends upon the time as well. So you can also have, like, this is the flight tube, this is the reflection mode. So you can also add one of the reflectors so that it will get more time to get separated. Here, here is the detector, so it will directly get detected here. But if you have the reflector mode, then the detector is here. So it will the ion source will come here. The samples, uh, the ionized samples will come towards this direction. Then after hitting that the reflector, it will go towards the detector. So now it will get more time to get separated and much more specific, better separation and analysis can be done using this technique. This is the formula. So T is for time of flight, M is for mass of the ion, Q is for charge of the ion, B is for accelerating potential, and L stands for the length of the flight tube. So this length will vary if you are using reflector. So the length will be combination of these two, and if you are using this detector, the length will be this much. So as I said, for sample preparation for mass spec, once you do the image analysis, you will cut the specific sample, you will do the gel digestion, and add it into the specific enzymatic digestion process. So for example, trypsin, the trypsin gets digested at arginine or lysine at the C terminal position. Then it will make certain fragments as per the lysis method. Then the digestion protein uh, will be further purified for removal of salts, buffer, etc. As I said, it will have certain gel part as well. You need to remove all of those parts, and your protein, your sample should have only protein. Then you will add the specific buffer to it, or specific matrix, the matrix itself, so that you can now add it on the matrix plate, which is called as Maldi plate. So that plate has your samples now, that is your analyte. Now laser will come in contact with this specific plate, and that will be getting ionized and then separated as per mass to charge ratio. Then when it detects, when it hits the detector, then in the software you will see certain peaks accordingly, and if the intensity is greater, the peak height will be greater. If the intensity is smaller, the peak height will be smaller. So this is how the protocol is. You will digest it. Once you digested it, you will add it in the sample and on the matrix, and it will get ionized and detected by the detector. So now in gel digestion reagents, these are certain reagents. This is the protocol. You need to destain it first, then you will do dehydration, then you will do reduction, alkylation, dehydration, digestion again. So this is the protocol for in gel digestion. So the spot you are seeing that is being stained, right? You need to destain that first. Then you need to dehydrate the sample. Then you need to reduce it again using DTT. Then you need to alkylate it so that whatever is the disulfide bonds which are broken, then they are again attached with certain groups so that they are not reforming again. Then you need to dehydrate it and then add the digestion buffer like trypsin solution. So this is how the protocol happens with DTT and then with the iodoacetamide, then with the digestion. For example, for trypsin, it digests at lysine and arginine. So this is how the dye looks. Once you wash it, it becomes transparent. This is destaining it. Then you will do reduction, alkylation, then digestion. So this is the whole protein. After arginine it gets, so it will form different fragments like this. Now this can be visualized in your mass spec. Then you will filter it using zip tips. So this is how the zip tip looks. So this is how the filtration takes place. The impurities will be washed away and your protein will be present. And This is a protocol what you are generally using for analysis. Then matrix selection also depends. There are different types of matrix available for mass spec. So this matrix is used for such kind of peptides which are less than 5,000 daltons. Similarly, this is used for higher than 5,000 daltons. So this is the matrix basically, which are finally resuspending it after ingel digestion. So for nucleotides, this is the one. So this is how different digestion matrix are available for Malditov. Then you will add the samples like this. Each spot will have your each sample. Then you will set the parameters in Malditov machine, and then you can run the sample, and you will. That's how the peaks will be generated uh, of different specific molecular weight, but different specific mass to charge ratios. Then using this mass to charge ratios, different uh, mass to charge values, you can come to a conclusion that this might be the specific protein which is present using the standards. Okay. Then this is the application. You can have it either backward approach or the forward approach. Forward is after the clinical trial. This is a backward approach. Initial identification for biomarker identification, etc. Yeah. So I couldn't do the questions as such. I think you all can go through the questions as we discussed during our week 1, 2, 3, 4 lectures. I also have like added the recordings of it. Maybe you all can go through that. But I think revising all 4 week lectures within 2 hours, I think you got uh, good revision of it. If I'm not wrong, you all can discuss with me accordingly. Yes, are you all there? Thanks, Aditi. So I think your exam is on which date? 21st. Okay. Okay. So I could revise the whole thing. I actually thought of doing certain questions also, but the syllabus itself is so huge that I could complete only the revision part of it within 2 hours. But if you want to put the questions again, you all can go through the videos once. Yeah, thank you. Okay, then all the best for your exams. Thanks, Aditi, Lekha, and Brunalini. Okay, so do you all want to ask me anything specifically, or we can stop the lecture? If not, then yes, thank you for attending, and I hope your exams will go well. All the best. Do well. Thanks. I think you all are given. You all are having my email ID as well, if I'm not wrong. I think that there might be a sheet shared with you all for telling you all that this is the TA for. I've just added it in the chat box. Okay, okay, fine. Yeah. 1402601 6 at So if needed, you all can ask questions over there. Thank you.